Yes.
Okay, so I'm getting a bunch of yeses. That's good.
Yeah, yeah.
Okay, so Kara is going to be recording the problem session, and so Kara, do you have that started now? And I hope the answer is yes.
Yes, we're good to go.
Okay. Cool. Just so that I can have that off of my screen. I hope you guys are all doing well. I know this is kind of like a weird setup because usually we're in a classroom, and lo and behold, we literally can't be in a classroom right now. So we're just gonna kind of go as we will, and any feedback you guys have, if you want us to like pause and like go through something a little slower, if you want to type that in the chat. Kara's gonna be monitoring the chat, and I'll be writing on the worksheet with you guys. So we're gonna go through that, and any feedback you have, this is our first week doing this, so we can only hope that it gets better from here. So let's go through the worksheet, and I'm gonna switch to tablet mode so you guys can all see my screen, right?
Will you share the annotated worksheet after we're done?
I have to check with Dr. Jones and Dr. Hallweg if we're allowed to do that, because we usually can't give you our copy to begin with.
Okay.
But I think you guys should have a blank copy available on the Moodle website, correct?
Yes.
Okay, so if you want to fill it out at the same time that I am, and then we'll do a check on seeing if we can share the annotated version that we have with you as well.
All right, thank you.
Yep. Okay, so we're going over translation this week. I believe that's been covered in your video lectures already, and a little bit before spring break too, possibly. So just kind of as an overview, what happens is you have this mRNA that's been created through transcription, which is a process we've already talked about. And then the ribosome goes and attaches to that mRNA and binds to the Shine-Dalgarno sequence in prokaryotes or the cap binding proteins in eukaryotes. So it goes in and it's looking for this specific signal, and that specific homing signal is going to help it know where to start and what it's looking for structurally. So it homes in on that, attaches to the mRNA, and then it starts doing what's called translating. So it's translating this mRNA code into an amino acid code that comprises proteins. So kind of the way that I like to think of it is you go from DNA to RNA, and it's called transcription. And that's kind of like you're taking something and you're just writing it down. So like if you were in a temple in like Egypt or something, and you're in this pyramid and you go in and you get the hieroglyphs and you just take a recording of what those hieroglyphs are, then that would be the transcription part of it. And then you go back to your library and you go and you say, okay, now I'm going to look at a book and it's going to tell me what I'm going to use and translate that into the language that I can speak and write in. And so then you take those hieroglyphs from that ancient language to a modern language that you can actually use. So that would be your translation, kind of to make it related to some kind of metaphor. So we're going to go and we're going to have these codons, and the codons are essentially each hieroglyph. So a codon is going to be a series of 3 nucleotides in the mRNA, and that's going to tell the ribosome that it's looking for a specific amino acid. A tRNA, or transfer RNA, carries the amino acid to the ribosome and then transfers it into the growing peptide chain. And so it's going to be using that based on the codon that's in the mRNA, is going to tell what tRNA should go with it. So the tRNA is going to have 3 nucleotides called the anticodon. So you have your codon and then you have your anticodon, and they're going to base pair together using that complementarity that we've gone through in other cases, such as the 2 strands of DNA binding together and the writing of the mRNA itself. So to start translation, the first thing that happens is that there's going to be a tRNA, and it's going to bind carrying this methionine, and it binds to the codon AUG. So that's always going to be our start sequence. So AUG is always going to be the first codon in a protein. And so it's the initiation codon. That gives it some specificity so that then you know exactly where the ribosome should start. So the ribosome looks for that site and then it starts there. And then at the end of translation, you have these release factors that'll bind to the termination codons, which are UAA, UAG, and UGA, and that'll cause the mRNA and peptide chain to be released from the ribosome. So it does this binding, and then you have a release factor that's here, and it tells it, okay, don't grow the chain anymore, we're stopping here. So that's how translation is going to work. So what we want to know then is we have this coding dictionary here. And when you have this coding dictionary, you have some chains that'll specify the same amino acid. And so you have these two here that'll both specify for phenylalanine, or Phe. So then each of these, some of them have more than one, and some of them look like they only have one. So here, trip only has one, but arginine has 4 that we can see right here. Make sure you pay attention, because arginine has 4 here, but it also has 2 down here later. So when you're going through one of these tables, make sure that you're paying attention to the whole table and not just assuming that because you found one place that you see that amino acid, that you've found all of them. That's kind of a common hiccup that people have. Okay, so then what we want to know is that we have this amino acid, tryptophan, and so we want to list the possible codons and their corresponding anticodons here. So we're told that this coding dictionary for our codon is going to have UGG for tryptophan. And so we're going to have the codon 5' to 3', and we're always going to write it like this. And it's going to be UGG. And so if this is the codon, the anticodon is going to base pair with it, complementary and anti-parallel. So we're still going to do complementary, anti-parallel. So the anticodon is going to go 3', 5', and then we're going to have C, C, A. So that's how you're going to figure out which one is the codon and which one is the anticodon. Okay, I know I'm probably moving like semi-fast with all of this, so if I need to slow down, like please be typing in the chat or something and tell Kara to make me stop or something. Okay, so then there are 61 codons that can code for these amino acids, but there's actually only 31 to 41 different tRNAs. And so some tRNAs, as we saw in the table previously, can pair with multiple codons because there's this relaxed base pairing called wobble rules at the 3' end of the codon. So this 3' end is called the wobble position. Because if you look up here again, and I'm going to use arginine. So on arginine, we have this CG, 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 but then the codon at the 3, or the base at the 3' end doesn't change what amino acid we're coding for. So we could have a G, we could have an A, could have a C, could have a U. Any of them work. And so that's the wobble position. It's going to be at the 3' end of that codon, so make sure you know that. So that means that a single tRNA can transfer the same amino acid to the growing peptide with a number of codons because you can still have that fit based on the wobble position that's down here. So now we're going to use this table here to answer the next few questions. So where in the codon does the wobble base pairing occur? And it's going to occur at the 3' end of the codon. So then for the next problem we have, for the following tRNA anticodons, list the possible codons each could pair with and their corresponding amino acids below. So we have a anticodon, which is 3' AAU 5'. So then we want to write, if this is the anticodon, then the codon has to go 5', 3', and the first part of it has to be U to base pair properly with this section here. But the 3' end of the codon is the wobble site And so then we have to ask which ones would be able to be in the wobble position to pair with this. And so we can go back up to our table here and we see that we have a U in this part of the anticodon. And so if we have a U in the anticodon, that means we can have an A or a G at this end of the codon. So we could do 5' UUA or 5' UUG. And then we want to know what amino acid we're going to be using. And so I'm going to go and I'm going to look for UUA and UUG for the codon. And so first position U, second position U, and then third position is going to be A or G. And so we can get leucine or LEU. So this is going to be leucine. Okay, so now in the next problem, we're given a codon. And so the codon that we're given is this 5' AAA. And so we want to list the possible anticodons that you could have base pair with it. So if we have a AA, then we're going to write 3', 5' here for the anticodon. And we know that the 3' end has to have U in order to base pair with these two A's. And then for an A to be here, we would have to have either a U. I'm going to erase this so we can see it. So we have an A in the codon. So we would either have a U or an I in the anticodon. So we can have this U, or we can have 3' to 5' U, U, I. So the first time we were moving in a direction from here to here. This time, since we were given the codon, we were moving from these A's over to the U and the I in this direction. And just using that table. Okay, so now to do the next one, we have this codon here, and then our anticodons are going to be 3' to 5' again. And this time we're gonna need a U and a U because we have two A's here again. But now in the third position, we have a G. So now on this table, I can have a G there or there. And so that means my anticodon can be a C at the end, or if I write the other one, can also have a U at the end. Okay, so then now if we know that there's only one tRNA that's charged with lysine in a cell, which one of the above anticodons would you predict to be found in this tRNA? And so we're going to predict that we would expect there to be the one that is going to be 3' U, 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 5' anticodon. And the reason that we're going to predict that is that this anticodon, since it would have a U at the end, can actually match both of these situations, so both A and B, rather than only being able to match one of the two. So if you add this codon here that ends with the G, or this anticodon that ends with the C, then you would only be able to match the G. And so you want to be able to have the one that is going to match more because it's more generalizable, essentially. That's kind of one of the more confusing parts of this whole translation and wobble base pairing. But you would expect the one that's going to be more generalizable to be the one that's present, because then the cell doesn't have to expend as much energy making these tRNA anticodons. Okay, so the next section that we're going through is on the coding dictionary and the universal genetic code. So we have these codons, and they're going to specify the same amino acids in nearly all organisms. So to conform to this code, the tRNA can only base pair with codons that code for the same amino acid. So we want to know, does the tRNA anticodon 3' UAU conform to the universal genetic code? So we ask the question, what codons can this anticodon bind to? And so if we have a anticodon here that's going to be 3' UAU, then we're going to have codons that are going to start with 5' to 3', and they're going to have to be AU to start. And then this is our wobble position here. And so if we go back up to the table that we have up here, we have a U in our anticodon. And so then we can have an A or a G at the end of this codon. And then we could also have as our other codon, AUG. So I've underlined the wobble position in here. So then to answer the question of, do they conform to the universal genetic code, we asked, do all of these codons code for the same amino acid? And if the answer to that is yes, that means that it would conform to the universal genetic code. So we have AUA and AUG. So what I'm going to do is I'm going to go back up to the table that we had to start out with. So I'm going to go back up here, and I'm going to look for the codon UAU. So U, and then I'm going to go to A, and then I'm going to go to U. Okay, that's tyrosine. And then I'm going to go and look, or, oh wait, this is supposed to be AUA, sorry. Getting them mixed around. So I'm going to go to A, U, and then A, and that's going to be isoleucine. And then I go and look at AUG, and that's methionine. So we have this one codes for ILE, and this one codes for MET. So then the question is, do they code for the same amino acid? No. The first one is going to code for isoleucine, and the second one's going to be coding for methionine. And so that means that the tRNA does not conform to the universal genetic code. Okay. So then the next thing we're going to do is look at this problem where we're working as interns at a wildlife preserve in East Africa. And so we're looking at these monkeys called African vervet monkeys, and they have become ill with these rashes and lesions. So you look at the pictures that you've seen in books, and actually this looks quite like the chickenpox. And we know that chickenpox is a double-stranded DNA virus. So we're able to isolate DNA from the virus and have it sequenced, and so then we can determine if this simian varicella primate chickenpox is the same as the chickenpox that we see in common disease. So we need to look at the genes for this. So what we decide to do is that we look for genes for the viral protein coat, which is an important membrane fusion protein during infection. So then we know that glycoprotein K in the human chickenpox virus is 340 amino acids, and it begins with the sequence MQAL. And so we think that if they're the same protein sequence, so if they're similar, then glycoprotein K in this virus would have a protein sequence that would follow similarly this MQAL pattern. And we have some DNA from that region of the genome. So let's try to find the reading frame for glycoprotein K in the primate virus then. So the first thing that we need to do is try to find the start site and the reading frame. And so we need to figure out which strand that we're given here in this double strand. So this is strand 1 and this is strand 2. So which one is going to be the template and which one is going to be the actual coding sequence. So what we're going to do real quick is we're going to write them a little bit further. Well, actually, we don't need to write them further apart. This will make it easier so you don't have to watch me copy the entire thing down. So what we're going to do is we're going to look for potential start sites because we know we have to start with AUG as our start site no matter what. And we know that it could be on either strand. So if we know that we're going to have an AUG, well, here's an ATG, which in the RNA would be AUG. And then let's look for some other ones. Here's another one here. And then let's look at the bottom of it. So if we're going this way, the other way, we could get an AUG there. Here's an AUG. Okay, so we have a couple of AUGs in here to look like. So if we look at the first one and we take this as our sample to start with, this would code for M. And so that's methionine. And so if we're going this way and we're reading that way, the next codon is TTC. And you would want to look in your codon table for that. I'm not going to because I have the key sitting in front of me, so I don't need to be scrolling back and forth the whole time. But you're going to look in the codon table, and what you're going to find is that this codes for F, and then the one after it would code for C. And we know that the start of the sequence is MQAL, so kind of looks like that's not going to be the correct start site for us. So that's not our start site we're looking for. Okay, so then I'm going to move down here and I'm going to do something similar, and we're going to be going that direction again, and we're going to start with methionine, and then the one after that is going to be V for valine, and then the one after that is going to be L. And that doesn't look like it's the right sequence either. So this is kind of a trial and error type thing. And so then if we were to go the other way from here, so say we had started at this sequence right here, I'm going to draw in another color real quick. So now we're going this way from this start site. This guy's M, this one is V, the one over here is R. So that also doesn't look like the right start site. And if we go down here and we know that we'd be going this direction, then we're going to start with M, and then the one after that is going to be Q. The one after that is A, and it looks like we're on the right track. Let's check the next two. We get L, and then the one after we get G. So it looks like we've got this MQAL start, and so the strand that's going to be the coding strand is then going to be this bottom strand. So this is the coding strand, because that tells us the actual sequence. So then we're going to have the top be the template because the bottom is coding. And then we have to ask the question of what direction does transcription proceed? And that's part of the reason I drew these arrows on here. So I'm drawing the arrows there, and so we know that transcription is going from right to left.
Hey, Anna.
Yeah.
Can you go over one more time how you figured out what the correct start site was?
Yes. Which part of it do you think would help to go over?
Um,
Sorry, just because I went over 4 different tests for start site.
I would say the bottom one. Actually, hold on. Let me make sure.
And thank you all for being patient. I know that like this transition to online stuff is kind of weird.
Okay. Well, we'll see what the
So,
response is.
okay. So to go over why we chose this as our start site. So we're given the sequence up here. We're given that this start sequence in this human chickenpox virus is MQAL. So that's the sequence that we're looking for the whole thing. And so when we start and go through these different ones, the first one we get MFC, the second one we get MVL. This one is going the opposite direction. We get MVR. And then we go down to here, which is another AUG or ATG in the DNA, but it would be AUG in the RNA. So then this is a start site that would be M, and then we get a Q, which matches with the Q we should have here. And then an A matches with the A, and then an L that matches with the L that we should be seeing. So that's where we get which part is the one that's going to be the coding area.
I think that covered it.
Okay.
Thank you.
Yeah. And so then the mRNA and the amino acid sequence that we're going to write out is that we start with that AUG. Sorry if that's a little repetitive, and that's for methionine. And then we're going to have CAA, and that's going to be Q. And then we get a C, or yeah, G, C, G. That's going to be A. And then a CUG, and that'll be L. GGA, and that's going to be G. AUU, and that will be I. A for K. ACA for T. GAA for E. And then I am bad and wrote too far to the side, so the last one I'm going to put down here, but it is still part of the same sequence, and that's going to be for H. So then our protein sequence is the bottom. So this is going to be our protein amino acid sequence, and this is going to be our mRNA here. And I separated it for you guys by codons so you can see those triplets and how they're corresponding to the amino acids. Okay, do we have any more questions for that for right now? Or are we good to go?
We just got a question. It says, for the coding strand, is the 3' end on the left and the 5' end on the right, since we're dealing with codons and not anticodons?
Can you repeat that?
Can you label your 3' and 5' ends on the strands?
Oh yeah, sorry, I forgot to do that. I always forget to do that in this one. So this is 5' and 3', 5' and 3'. And that's because this here is the template, and this is our coding strand. That's how I know that this end is going to be 5', because I have to have a 5' methionine here on the coding strand, and then you go 5' to 3' and synthesize that way. So it's all about which way we're doing transcription in. Does that answer that?
Yep, that answered it.
Okay, sweet.
Are there any other questions before we move on?
Okay, if there aren't any, I'm going to move to the next page.
Okay, yeah, it looks like we're good for right now.
Okay. Okay, so now we're going to go into a thing about glycoproteins. We know that these are important for this viral entry and fusion, so we're still looking at this chickenpox type virus. And we want to be able to further study the glycoprotein K in this virus. So what we're going to do is translate the full gene region. And so here we're given a portion of the template strand of DNA. So this is the template, not the coding. And so what we want to be able to do is take that template strand and assume that the first codon for the peptide begins with the first nucleotide of the portion of the RNA. So then that there's no introns. So we're not going to start at AUG, but we're going to write out the mRNA and the amino acid sequence here. So this is the template. So if I want to write the mRNA, I'm going to write it up here instead of down below in the blank. So here's my template. And I know that my mRNA is going to be anti-parallel to it and complementary. So here's my mRNA here. And so for my mRNA sequence, I'm going to get G, U, A, G, C, C, U, G, U, 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 G, G, C, A, U, A, U, C, A, G, A, and U, G, A. So I'm just getting that mRNA sequence by flipping across and using the template to translate it or transcribe it across. So if I want to write that again really, really fast down here, I have G, U, A, G, C, C, U, G, G, C, A, U, C, A, G, A, G, A. Okay, so there's my mRNA from 5' to 3'. And that is going to match with the N-terminal end of the protein. And then over here, we're going to have the C-terminal end of the protein, and that's going to be COOH. Actually, it should be 3 O's. Okay. So we've got an N-terminal end and a C-terminal end of the protein. And this is the same wording that's going to be used in the problems on your homework. And if you guys were taking a regular exam, it would be on there. But I believe you now have video exams, so I don't think that this will be the exact same type of wording. But just to caution you, this is the same kind of wording that'll be used on your homework. And so what we want to do then is just go up and be able to match these codons. So I'm going to do a few of them by actually going up to the sheet. So the first 3 we'll do from the sheet itself. And if we go back up to our table of codons, what we have here is our first codon goes G, U, A. So I'm going to go G, U, and then A. And so GUA is going to code for valine. Second codon that we have is going to be starting with A and then go to G. So I'm in this box. And then the last letter is C. So I'm going to get serine. So from A, G, C, I get serine. And then for C, U, G, so CUG is for isoleucine. Or not isoleucine, regular leucine. I can't read. Okay, so that's how you're gonna do all of those. And so from these ones, I got V, S, and L. Kara, can you like pop into the chat and see If anybody would like me to do a few more of those on the actual codon table, or if it's okay to just write them out and you guys understand that process well.
Yeah. So if you want Anna to write a few more, tell me now.
Yeah.
Everybody good? Okay.
Okay, so radio silence on that one.
I got a no thanks and a yes. That's fine. I'm not sure. Maybe do a couple more.
Okay. I mean, I can. I'm going to skip the middle ones and do the end ones because the end ones are a little bit more interesting. So we're going to have an F here, a W here, and an H. And then for the last three, I'll go back up to the table and do those real quick because they're a little more interesting than the middle section is. Sorry, middle section. Okay, so I have an AUC that I need to take care of, an AGA, and then a UGA. Okay, so for AUC, the first position, so 5 prime, 3 prime for each of these. So 5 prime, we're going to have A, and then a U, and then a C. So that's going to be isoleucine. And then for the next one, we're going to have A, and then G, and then A. So we're going to have arginine, which is R. And then UGA, so U, G, because you're always going to look first position U, second position G, so I now have to be in this box. And then for UGA, we have this codon here, and that is going to be stop. Important note about a stop codon, there isn't actually a amino acid called stop. It just means that it ends there and that there won't be anything else added on. So stop is not an actual amino acid. It's just telling you that that is the end and that you're going to stop reading and stop trying to translate. Okay, so back to here. We figured out this one was isoleucine. This one was going to be arginine, and that this is our stop. And so there isn't actually an amino acid that gets put there. It's just telling the ribosome to stop reading and cut off the protein making there. Okay, does anybody have any questions about how to go through a problem like that?
No, I don't think so. Is everybody okay to move on? Okay, yeah, everybody says they're ready to move on.
Okay, sounds good. I just want to check before moving on really fast or something. Okay, so the last problem that we have on here is that we're going to compare the simian virus DNA to the human virus DNA. And so when we do that, we actually notice some differences. And so we have to ask the question of, yes, there's differences in the DNA, but does that mean that there's going to actually be any difference in the protein? And then to answer that question, why or why not? So first we want to look and see where those differences are. So we're given that the simian virus has an AAA here, and the human has a GAA. So these two are different. And then we're given these next ones, and so these ones look like they're the same, so we can kind of ignore those for right now. These ones look the same, so we could ignore them for right now. And then it looks like these are going to be different. So those are the differences that we see in the template strand. So then what we want to ask is, what does the mRNA look like? So for the simian mRNA, we're going to write it 5 prime to 3 prime. And so we have CAT, which is going to end up being GUA to start out. So it's coming from here. And then we have TCG, which is going to be AGC. Then we have CUG. And then U, U, 3 prime. And then for the human mRNA, we're gonna have 5 prime G, U, C, and then A, G, C, C, U, G, U, U, C, 3 prime. So to highlight what the difference is between the two, here I have a U, here I have a C, here I have a C, here I have an A, and notice that all of these differences are in the wobble position. So if they weren't in the wobble position, we would know that they were going to code for something different automatically. But because they're in the wobble position, now we have to check what codons we could get for these different anticodons, essentially. So we're going to go and we're going to look at GUA. And so I'm going to go back up to my table at the first page. So we're going back to here. And so I want to look at GUA and GUC. So if I go to GUA, G first codon, U second part of the codon, and A. So GUA is going to be for valine, and then GUC is also going to be for valine. So for that first position, it looks like they're going to be the same. So they're different codons, but they code for the same amino acid. Then for that second place that we saw a difference, I'm going to go and I'm going to look for U, U, U. So U for the first one, U for the second, U for the third is right here. That codes for phenylalanine. My other one was UUC, and that also codes for phenylalanine. So what I'm going to write down here is that we have these two, which are GUA and GUC, both code for valine, which is V. And then for these ones here that we're matching up, both of these code for phenylalanine, and that's going to be F. And so they're two different codons for each of them, but they both code for the same amino acid. So in the simian one, you're going to have valine here. In the human one, you have valine. They both code for V. In this one, there's the difference in the DNA still, but you have a U here and a C here, but at the end of the day, they both code for phenylalanine. So at the end of the day, the protein is going to be the same. So the different codons code for the same amino acid. So even though the DNA sequence is different, the protein sequence is the same. Okay, so we've been able to answer the question that we were given up above here. So we asked if the protein is going to be different, why or why not? So the protein is actually going to be the same because even though you have a difference in DNA sequence, you have these different codons that are going to code for the same amino acid. So at the end of it, the protein sequence will be the same between the two. So anybody have questions on that?
It doesn't look like it. We'll see if any come in.
Okay, I mean, we have some time, so like if people want to process for a minute and we can just pause and wait for a few questions, and then if people don't have questions, we can move on to just giving you guys the problem session question.
All right, let's do that. We'll give you a minute to gather your thoughts and ask any questions you might have.
Yeah, just in case anybody's typing something.
And I know we had a question asking about the mini videos for the exams, so we'll go over that at the very end.
Okay, am I good to move to the problem session question, Kara?
Can you go over wobble one more time? So we had one question that asked if wobble positions were also able to change according to the anti-codon-codon base pairing rules, and just a general question to go over the wobble position one more time.
Okay, yeah, let's go over the wobble position one more time, and then maybe that'll answer the more specific question at the same time. Okay, so the wobble base pairing rules essentially say that there are multiple anticodons, or an anticodon can pair to multiple codons itself. So you have multiple different codons here as far as what they end with that can pair with an anticodon that would have U at the 5' end. So the idea behind that is that you could have one codon here, and so if you know that you have some codon that's going to be 5' to 3', then as long as you have two U's here, if you have an A or a G, you can have the same anticodon that's going to pair with both of these codons, and then you would get leucine out of it. And so then the same thing would go down here, where all four of these have, for their first two positions, which the first two positions I would call rigid if we're calling the last one wobble. So then in this rigid position, you have C and U, but then at the end of this, you can have any of these and you're gonna have a codon for leucine. And so this is the 5' end of the codon and the 3' end of the codon. So the wobble position is for that 5' end of the anticodon as well, which is why in this table here, this is the base at the 5' end of the anticodon, and then the base in the corresponding codon, which is going to be at the 3' end of that codon. Does that answer their question?
I think it answered yes. We got a yes and a yes.
Okay.
So we've got a couple that want you to go over translation practice problem 2. Basically, wherever you labeled your 3 and your 5' ends, if you could just briefly show those one more time and kind of talk through how you figured out which was 5' and which was 3'.
Okay. So the way that I figure out for this problem, which is going to be 5' and 3' for the ends, I know that it's the first problem on here, or the second problem on here to do that, part B. I think it's easiest once you kind of have your orientation as to which strand is going to be your coding and template. So strand 1 and strand 2 here are going to be your top and your bottom. So if I erase these real quick, just so we can kind of go over it from a little bit more of a blank slate. So the first thing that I wanted to do was determine the start site in that reading frame. And so the first thing that I went through and did was find all of these start sites. So all of the start sites are going to be AUG, so I find all AUG. So I find all the AUGs on each strand going any direction, period. So that means that they can be going whichever way works. So that's where I found, you know, this one going this direction. So this one going from left to right, this one going from left to right, and then this one going from right to left, and this one going from right to left. So I go and I find all of those codons to start off with. And then the next thing that we did was figure out the sequence that would be following it. And what we're trying to do is find a match for the sequence that we've been given, which is MQAL. And so we went through and we were able to say MFC, MVL, MVR, and we were like, okay, well, none of those match, so those aren't the start site. And then we go through and we find this one that's MQAL. And that is the actual sequence that we're looking for. And so we know that this has to be the coding strand. And that code has to go 5' to 3'. So we know that we're on the coding strand for this one because we see the actual protein sequence that we're looking for, which is that MQAL. And so we know that that has to go 5' to 3'. So then I know that this strand starts 5' over here in order for this to go 5' M to 3' L, as far as how it would have to be for the mRNA, which means that this is going to be 3' over here. And then since we know that the two strands of DNA are going to be anti-parallel, then we know that this has to be 5' and that has to be 3'. This we know is the coding. This is our template. Does that clear that up?
It looks like it did for at least one person.
Okay.
Okay, I'm getting some other yeses.
Okay, hopefully that explanation helps. It's one of those things that, like, I'm sure Kara can attest to this too. After doing this a million times, like, you can just kind of look at it and we just kind of know. But for you guys, you know, take your time and, like, think about it in practice and everything. It's somewhat automatic once in a while.
So somebody asked how you found the AUGs.
So the AUGs are going to be anywhere there was an ATG in the DNA. So I'm just looking for all of the, what would be ATG in DNA. And so I know that this is going to be AUG in an mRNA. This will be AUG. This will also be AUG and AUG. So I kind of like to go through and look for all of the ones going left to right first. And then after that, do a back sweep and go right to left on each strand afterwards. And that's basically how I find them, is I just go and look in the sequence for ATGs and know that that's going to be where the AUGs are. So all of my AUGs are going to be in this green color. And that's all of them that are in the sequence. Okay.
All right. So I think that's good. Yes, there is a participation question for this problem session. We'll get to that in a minute. And then we have one last question. We wrote the mRNAs, the mRNA sequence for each virus strand on the last page.
Yep.
Are these the codons? So what are the codons here that you're?
So each one of these, this is a codon. So this is one codon. Here is another codon. And then we have a third codon. And this is the fourth codon.
So the anticodon is what would be on the tRNA.
Yes. And then as a like separate note for this is a codon table. So this is going to have the codon itself. So this is not an anticodon table. This is a codon table. So the sequence that you see here, 5' to 3' is the codon.
Okay. So I think that's it for the questions. If you have further questions, you can email and we'll try to clear them up for you. Just to go over mini videos really quickly, if you look on Moodle, under your exam mini videos, there are prompts for you to answer. And so what you would do is you would go in and you would choose one of the following prompts, and then you would follow the instructions to upload it and go from there. So just remember to make sure that your videos are within the time limit that they're supposed to be. Dr. Hallweg sent out an announcement earlier today. So if there are questions, feel free to reach out and ask. And if there are no other questions, I'll let Anna go ahead and assign you your participation question for this week.
Yeah, and the one other thing too is I think they have a rubric for their videos that should be available on Moodle. And so, you know, if you're looking at what kind of grade you want to get, make sure that you look at like that rubric itself so that you can kind of gauge, you know, is this, you know, getting the creativity points? Is this getting the correct content points? Because they did say that, you know, you could get a good amount of credit on full content, but if you were very uncreative and, you know, just sat in front of your computer and talked for 3 minutes, that we're supposed to dock you on creativity. So it might be helpful to know that. Maybe you don't care.
Yeah, and on your rubric, that is under, if you are on Moodle, under the exams column or tab section, there's a PDF for exam or Genetics 311 exam mini video information. If you click on that, that will have your rubric, that will have everything that you need to know.
Okay, so then for like the kind of last order of business and everything, the problem session question you guys are going to have is this question number 1 that I am on. So it says, for the following amino acids, list the possible codons and their corresponding anticodons below, and that's for tryptophan. Make sure that when you're doing this, you label the codon and the anticodons so that we know which one you are giving us for each one, because you're going to give both, but make sure that when you're doing that, you're labeling and saying, here is the codon, here is the corresponding anticodon for it. Do you guys have any questions about how to do for that?
I don't think so. As far as uploading, or as far as your participation, we're in week 9. And if you click on it, it's just the one section. So it's not like when we were doing this in class and you had to go to your session number. This is just all one thing. Just go to the quiz, put in your answer, and that's it.
Yep.
And you're just going to answer exactly what Anna has written here for number 1. So the codon and anticodon and what those nucleotides are. All right. If you have any other questions, you can reach out. But I think that wraps it up for today.
Yeah. Have a good week, you guys, and, you know, socially distance, but still try to enjoy your life. Go for a walk.
Yep, the rest of the week looks beautiful. Yes, the star. That's your question. All right. Bye, everybody.
Bye. Have a good day.